All right, AP Bio, welcome to the cell cycle. This is the second part of Unit 4. And so the plan is once we get through Unit 4 next week and probably into the week, 2 weeks from now, we'll definitely have a Unit 4 exam over everything. So this is cell cycle. Hopefully this is a review from if you took honors bio. I'm going to have to try to move so I'm not blocking that image right there. So the cell cycle, it's the life of a cell from its formation until it divides into two cells. And so essentially we're going to be talking about mitosis. And so function cell division is reproduction, growth, and tissue repair. So the genome, again, as a review, is all of the cell's genetic info. And so in prokaryotes, as we've talked about, they have a single circular chromosome. And then in eukaryotes, they have more than one linear. So for example, there's 46 chromosomes in humans, mouse have 40, fruit flies have 8. And so each chromosome must be duplicated and duplicated before cell division. And so a duplicated chromosome is two sister chromatids attached by a centromere. You should remember that. Regardless, that is one chromosome. It's made up of two replicated chromatids, which we'll talk about. And so when we talk about somatic cells, those are body cells. So there's their diploid, 2N, two of each type, and they divide by mitosis. So humans, 2N equals 46. Gametes or gametophytes, that's your sex cells, and so that's sperm and egg, and they're haploid and one of each, and they divide by meiosis. And so humans, your haploid number is 23. And so here's our phase of the cell cycle, G1, SG2 interphase, and then you get into mitotic. So I'm just going to have to keep moving my face around here. Sorry, I'm not going to start this over. So the phase of the cell cycle, we're going to do something in your bill, but it basically goes G1S, G2, mitosis, cytokinesis. Where those boxes are should be arrows. Interphase makes up 90% of the cell cycle, with G1 is cell growth. S phase is DNA replication, which we'll review. G2, prepare for cell division. Then you go to M phase, and you have mitosis and cytokinesis. Oh boy, this is going to get old real fast. Let me see if I can't change the size of the screen. So anyways, you have to deal with it. So mitosis, PMAT, prophase, metaphase, anaphase, telophase. And so mitosis is a continuous process. The chromosomes become visible. They usually start as chromatin. They align at the equator. They separate, and then they form two daughter cells. And so there's prophase and prometaphase. AP exam's not going to focus on this. This is like introductory bio stuff. So then you've got your metaphase, anaphase. And then telophase and cytokinesis. And so cytokinesis basically is when the cytoplasm of the cell divides. And animal cells form a cleavage furrow, and then plant cells form a cell plate made up of carbohydrates. So cytokinesis in animal versus plant. As you can see, there's those images there. And there's your animal cell division. I'm kind of going through this quickly because this should be review. So G2, prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis, back into um, interphase. Plant cell division a little different. You can see the nice little boxes there. So which phase cells can you identify? If you want to pause this video and go through and count them up, be my guest. But there are some distinct phases. General hint, if it's usually a nice circle with a dark spot in it, probably like this one, that's going to be interphase. Cells spend most of their time in interphase unless they're cancerous. So again, there's an onion cell. I think it's onion cell, it's a plant cell. So during anaphase, chromosomes walk to poles by motor proteins. This is kind of important because these are where issues can occur. And then kinetochores, these are microtubules that shorten at ends as they depolymerize or they break down. And so this is kind of the idea. This is a misconception on how the chromosomes get there. They actually walk back. They're not pulled back. So I wanted to make sure that slide was in there. So bacterial cells divide by binary fission. So again, chromosomes replicate, basically splits in half. So this is the main point of what I want you to know for Unit 4 is this cell cycle control system, these checkpoints. So a control point where there's these stop and go signals, and they regulate the cell cycle. So these are the major checkpoints you know. G1 is the most important. Put a big star, highlight that. Controlled by cell size, growth factors, and the environment. So this is your go. So if it goes through G0 or go, complete the whole cell cycle. Sorry, not G0, that's a go. If it's a stop, the cell enters non-dividing state, which is your G0. So nerve cells, muscle cell state, G0. Liver cells can go back and forth. The G2 checkpoint is controlled by DNA replication. DNA mutation or cell size can stop it. And then the M phase is your checkpoint. Check spindle fibers, attachment to chromosomes at kinetochore. So here's your G1 checkpoint. Without go-ahead signal, cell enters G0. With go-ahead signal, continuous cell cycle. Here's your M, prophase and metaphase. It'll allow it to go through mitosis. And then, oh, my image didn't show up. But during the M spindle checkpoint, basically the kinetochore is a protein associated with DNA at the centromere, and my image didn't show up. So we'll try to get that fixed during class. So here's your internal regulatory molecules, and these are things they're going to ask you about. Kinases, which we've already talked about. There's these things called cyclin-dependent kinases, or CDK. This is a protein enzyme that controls the cell cycle. This is what you're going to see your kinases. When they're active, when they're connected to a cyclin. So a cyclin-dependent kinase, it's a kinase attached to a cyclin. And what cyclins do, these are the proteins that attach to kinases to activate them. So you can see the different levels fluctuating of these kinases, or these cyclins in the cell. MPF is maturation promoting factor, and this is a specific cyclin CDK complex that allows the cell to pass G2 and then go on to M phase. There are some also external regulatory factors. So there's anchorage dependence, density-dependent inhibition, density-dependent inhibition. So what will happen is, so I should go through the sequence here. So cells require a surface to do cell division, so they'll attach to a surface. Once they attach that surface, they're going to start dividing, and they're going to form a single layer. Once they touch the edges, they stop. And so what will happen is, as you can see, once those cells reach the edges, they stop. If you go in and remove some of those cells, they're going to grow until they touch the surface. And so in normal mammalian cells, they will stop. Cancer cells, they start to pile up on top of each other. So here's some external regulatory factors. You got growth factor, proteins released by other cells to stimulate cell division, density-dependent inhibition, like I said, and then anchorage-dependent. Cells must be attached to another cell. So cancer cells are different. Cancer is a disorder in which cells lose the ability to control growth by not responding to regulation. It's a multi-step process of about 5 to 7 genetic changes for a cell to transform. Often they lose anchorage dependency and density dependency, and so that's why a tumor forms, because they'll start to pile up. So some of these transformation is a process that converts a normal cell to a cancer cell, and when this happens, it can form tumors. So a tumor is a mass of abnormal cells, so a benign tumor being a lump, a malignant tumor is invasive, and so when it becomes invasive, it impairs function of one or more organs, and that's what's called cancer because it's moved. And when something metastasizes, it separates from a tumor and travels to other parts of the body. So normal structure versus cancer structure. So there's a normal cell. You can see the cytoplasm is different, nucleus, nucleolus, and chromatin. So typically in a cell, you've got large cytoplasm, a single nucleus, single nucleolus, fine chromatin. In cancer cells, you got small cytoplasm, multiple nuclei, multiple large nucleoli, and a coarse chromatin. It's a mess. So cancer cells have some abnormal numbers of chromosomes. Metabolism is disabled. They lose attachment of the extracellular matrix, so they can spread to other tissues. Signaling molecules cause blood vessels to grow toward a tumor. So there's all of these issues. So often we talk about treatment. You may have heard of some of these like surgery, radiation, or chemo. There's personalized medicines such as breast cancer. 20-25% tumors show high HER2 level receptors, and so they use Herceptin to block that protein because they block that receptor. So cancer risk factors, tobacco use, for those of you that chew or smoke, obesity is a factor, pathogens, unknown. You got physical activity, diet, occupational exposure, alcohol, reproductive factors, UV light exposure, environmental pollutants, prescription drugs. So we can't get rid of. I mean, you almost cannot eliminate any of your risk factors. So cancer prevention, anyone can get cancer, but there are ways to minimize it. Don't smoke legal or illegal, hookahs, chew, secondhand smoke, jewels, whatever. Use sun protection, exercise, and try to keep your weight at ideal. Eat fruits and vegetables if you can. Use screening preventative measures, checking breasts, testicles, mole checks, safe sex, abstinence, or using condoms, vaccines. Any of those will work for cancer prevention. And so here's some other cancer prevention recommendations. So here's a summary of your cell cycle. Interphase G1, SG2. You've got cytokinesis and mitosis. And so there you go. That was the notes in about 10 minutes. It'll take us more than that to get over it. But I just wanted to give you just a quick run through the notes. This is where Unit 4 wraps up, and then we can move into Unit 5, Unit 6, Unit 8. We'll be done. We've already done Unit 7. So again, hopefully you found this helpful, if you watch it or not. But this is me signing off from room 102 at Tiffin Columbian High School, Tiffin's beautiful South Side. I will see you in another video. Have a great day.